Welcome to the second part of the Chapter 8 video presentation for CJC 390. Alright, let's continue how we describe and record blood stains that we find. We describe the form, color, size, position, direction of splash. Obviously, photography plays a huge role. So you'll have blood interpretation experts come to a scene and look, but they'll also work with the photographs that they find with close-up views, and scale is extremely important for those folks to work with to determine the size and the shape of the stains that they're looking at. Sketch can be helpful too, and increasingly computers are aiding the process as well. How do we determine what we have is in fact blood? We have presumptive blood tests, also known as catalytic tests, that tell us if what we're looking at is some sort of stain that is not blood. Many things can be misinterpreted as blood or not. So presumptive blood tests can tell us at the scene if what we are not, whether we are looking at blood or not. Now you have to look and see that these presumptive blood tests are not conclusive, and that's the important thing to remember here. These tests that you do at the scene merely tell you this is likely blood. You have to collect that evidence, send it to the crime lab, and they will confirm that yes, it is in fact blood, and yes, by the way, it is in fact human blood, because often we find blood from animals too. Presumptive blood tests have to be handled with care because some of them are carcinogen. Just some of the examples, you don't have to remember all of these, but LMG, Kastle-Meyer, TMB are used. Depends upon the circumstances. Fluorescent is a type that's used similar to what many of you have heard of, luminol. It is a little different than luminol, but it is another type that you can squirt on an area that may have been clean, turn off the lights, and it will actually fluoresce. Luminol is one that many have heard about. It's been popularized through crime shows and things of that nature. It's where you simply spray an area with a reagent, you darken the room, and it luminesces. And what you see here in the middle slide and the left slide, pardon me, is an overlay. So you can see where the stain not only shows in the darkened slide, but also in the lightened room, and it gives jurors an opportunity to see where that stain presented. Another example of luminol that shows where an individual obviously attempted to wash blood off their hands in a sink, and it lit up the entire sink. In a bathtub, in a vehicle, you can see where they had blood on them. They got in a vehicle. It may not be immediately apparent when you first see it, but when you darken the vehicle, take some pictures, the luminol shows exactly what it was. Searching for blood sounds easy. It is not, because it presents in so many different ways based upon the age of the stain, what the surface is that is on, the color of that surface. But you can see it as fresh blood, obviously reddish brown and glossy. As it gets older, it grays, and it's very old stains. Well, the gloss will fade completely, and it turns very gray. Our old friend oblique lighting, pardon me, can help find blood, and so can different colors of lighting. So you'll often see crime scenes processed with different color filters on lighting. Places you might find blood include, think of the areas where you wouldn't see with the naked eye, perhaps, like drain traps. If you've ever worked on plumbing, you know what a drain trap is. It is the area that catches the water prior to dumping it into the sewer lines. And you'll often find blood in those drain traps, sinks, garbage pails, towels that they used as they clean themselves off. Obviously, clothes and weapons, and as we talked about, where gravity takes it into cracks. Knives, even if they were freshly used in a stabbing, may not show as much blood on them as you might think, because the wounds, if you look at the human skin, it's very elastic, and it tends to collapse around a knife blade as it is pulled from an individual, so it may, in fact, at least on the appearance of it, wipe most of the blood off, although you can recover that blood. There are still microscopic amounts that you can locate through various means. Blood is very fragile. It's one of the types of evidence we tend to work with quickly if we can at a crime scene because it is so perishable. So if we have an outdoor body, for example, we talked about slow and deliberate processing early on the course. But if you have a body that's outdoors and you see a thunderstorm rolling in, a severe thunderstorm, that's when you might pick up the pace, or you might try to handle that perishable evidence quickly because you know it's in danger of being disturbed by the rainstorm. The environment plays a big role, so blood is impacted by heat. That speeds deterioration. Now, on the opposite angle, refrigeration and freezing slows deterioration, and that is important to remember. That when we collect blood samples from individuals, for example, in a driving while intoxicated case or whatever we need it for, we refrigerate it because that slows that deterioration process. Obviously, again, we can find blood in many different forms, including liquid, damp, or even a dry state, and that makes a difference in how we collect it. Liquid forms, cotton paper, cotton-tipped applicators, filter paper, cotton gauze are often used, and we put them in an unstoppered test tube for later freezing. And dried forms, we'll scrape them off with a clean razor blade, scalpel, a knife onto a clean piece of paper, and fold that and seal it twice in an envelope and then in another envelope. We'll often also use moistened cotton swatches stuck between forceps to collect moist stains too if we're not able to scrape it. If we find stains on an object, it's important to photograph it before we try to collect the blood first, because again, we want to preserve the stain. We also want to be able to show the jury where the stain was, and it could be useful in interpretation of blood spatter analysis too. And in some cases, if we can take the entire object, we will. And in some cases, that may be in a vehicle seat, couch cushion, an entire part of a wall. It just depends upon the severity of the crime and what we're dealing with, how big the item is that we may collect and keep. With clothing items, what you don't want to do with blood, it's a moisture-based material, so you don't want to stick it into a plastic bag and air seal it, because what that will do is that will speed the development of mold, which attacks the blood. So we will hang those in what we call a drying chamber, allow them to air dry, and then we will package them. Just a cotton applicator, collection of some blood off a tennis shoe. If we can't cut out, or if we can't collect an entire item, so if it's like a carpet from a house, we'll cut out a sample, and we'll also cut out a control. So we'll go to where we know there was not a blood stain somewhere in the carpet and collect that. Specimen collection has gotten much easier than it used to be. If we're looking, for example, for DNA, a physician, a nurse, or a medical technologist is the only one who can draw blood. So some jails actually have nurses on staff, which is kind of nice because they can do it. But most police departments do not. So we can't draw blood from individuals in a police department jail unless we bring a doctor there. Easier way is to take them to the hospital, but we can collect it, and we do as a matter of routine, or what are called buccal swabs, and alternative blood samples, where we just simply swab the inside of the mouth for epithelial cells from the cheek, and that gives us the DNA that we need. So it's easy to do. It's like sticking a Q-tip in someone's mouth and then sticking it in a test tube and sealing it up, and you have their DNA. ABO typing has its value in forensic testing, again, because it's very much easier to conduct one of these tests than a DNA test. If someone is type A blood and we know the suspect's blood was a type B, we know we will not get a DNA match. This is not the same person. Your blood type doesn't vary. It is what it is, and it always is what it is. So ABO blood typing is very important and still plays a role. If you have someone type AB, you know you have only 4% of the population, so it's a unique individual. Species typing too. I've worked a few cases where we found a lot of blood in a truck, and come to find out it was deer blood. It was not human blood. So sometimes the blood you see is not human. And forensic DNA typing is something we look for SARS-CoV-2. Obviously, it's on our genetic makeup. It's also known as deoxyribonucleic acid. You may remember Gregor Mendel from your middle school science courses where he talked about how DNA, gives you blue eyes or green eyes or whatever it may be, and also changes plants and things of that nature. The Sam Shepard case was a case that is very interesting. Sam Shepard is, you can read up more about this case if you'd like. I would encourage you to do so. He was convicted of killing his wife, and he said it was a stranger that he had found and stopped an attack on his wife, is what actually happened. And interestingly enough, DNA evidence much later has cleared Sam Shepard. It is the case on which the television program and the movie The Fugitive were based upon. So in a very broad overview, we're not going to get really deep in DNA, but DNA is the basic parts of DNA include A, T, G, and C, where A and G are purines and T and C are pyrimidines. That's very tough for me to say, so I won't try it again. But these are found in the basic elements of blood, also found in seminal fluid, tissues, bone marrow, hair roots, saliva, urine, and tooth pulp, so we can derive DNA from a lot. First DNA case was in Narborough, England, back in the 80s, and there was actually a book written on this, where an individual raped and killed a young girl. And they actually DNA tested the entire town in an effort to find this individual, something that would be extremely controversial today. One individual declined to do so and actually paid somebody to go in and take the test for him, but later on they figured out what had happened, and he was in fact the killer. Challenges to DNA. It sometimes is not as simple as it seems, as degradation. Putrefaction is simply the bacterial attack upon DNA in blood samples that degrades the samples. So it's a real challenge to make sure that your sample isn't too degraded, although they are able to work with a much larger degree of sample today than they were even 10 years ago. They've made some amazing advancements as far as processing DNA. RFLP technology is simply a way that they have been able to duplicate variable number tandem reports, which they don't know what they really do or why they exist in blood, but the length of the VNTR region varies based upon an individual, so they can make comparisons in those lengths. PCR technology, that's polymerase chain reaction DNA technology. Again, it's like a Xerox copy of short segments of DNA from a specimen performed in what they call a thermocycler, which we have a picture of here. PCR allows us to work with even smaller segments of DNA. We have STR analysis too, which evaluates specific locations or regions or loci found on nuclear DNA and compare them to samples collected. mtDNA, this one's important to remember. You can actually use the maternal source as a comparison. So this is kind of the Maury Povich, I guess, DNA test, where we can say that this is the mother of this individual. We can actually do that through a study of the mitochondria of the DNA. So if you don't have a DNA sample from a missing person, you can collect their mother's DNA and use that as a profile to confirm or deny bodies that are located. That happens in policing. We find a body, we don't know who it is, and after exhaustive research, we might resort to testing known missing persons. And you may not have the DNA profile of that missing person, but you may have their mother, so it can be used for that. Smaller source material required, such as hair root shafts, too. Y chromosome analysis tells us the father, so this might be a Maury Povich type of case where we can say this is the father. Obviously useful in rape cases too, where it examines the male chromosome. CODIS is simply the APHIS for DNA. So if you have DNA profiles in the CODIS file, you can compare a sample you collected at, say, a rape scene to the CODIS database and see if any convicted rapist was involved in that crime. So it breaks down the DNA profiles where you can input them into a computer system. And there's a population file that is used for general building the population of the database and making it more accurate. There's a forensic index, and those are unknowns. So it may be a murder scene where you have an unknown DNA profile of blood, or again a rape or something like that, or a semen sample you don't know who it belongs to, and you stick it into CODIS, and hopefully someday somebody's entered into CODIS and it's a match. And that often happens through the convicted offender index. So most states, I believe all states now, require felons or people convicted of certain crimes to give up their DNA profile to be entered into a database to match potential unknown crimes. And it happens a lot. You hear the term touch DNA, and it's not an actual process, but it develops off of PCR. And essentially all it refers to is all we need is a very small sample size to compare to make a match. So all you need is about 7 or 8 skin cells, and that is very small. We use touch DNA in a lot of felon in possession gun cases. All they really have had to have done is touch that gun, and we can say with certainty that they possessed it at some point. If you're a felon, you're not even supposed to touch a gun. The Scientific Working Group on DNA Analysis Methods, or SWOGDAM, they set the standards for DNA and what should be required to call something a match. And so SWOGDAM principles are discussed often in court. You should have just waited.